Good morning, students. This is our chapter 11, Biotechnology Principles and Processes. Okay. So first of all, we will learn about that what is biotechnology. Okay. So biotechnology, this term means a technology used in living organisms to do any changes and to do modify anything in living beings. Okay. So means this technology generally used at that time when we do some changes or we modified any change in organisms, okay, any living organisms. Okay, so at that time this technology is used. Okay, so the principles and the processes. Okay, so biotechnology deals with the large scale production and marketing to produce and process using living organisms, cells and enzymes. Means in this process what we have to do is we have to deal with the large scales of production. See, we having lot of production according to the basis of marketing, right, and according to the product and according to the process that is using in living organisms. Okay, whether it is a cell or it is a enzyme. Okay, so this is the meaning of the principles and the process of biotechnology. So one of the most dramatic development in genetic began in the early 1970 that will undoubtedly continue to have revolutionary impact on biological fare. Okay, so see, I am telling you in a very easy language that what is the meaning. Okay, so what is the principles of biotechnology. Okay, so we have already seen that the process. Now we will talk about the principles. The branch of biology by which important and permanent changes are created, right, means the permanent changes and important changes are created in plants as well as in animals. Okay, so in which way we can increase or we can in which way we can do the createds and the changes. Okay, so by increasing, by decreasing, by manipulating, by transplanting of genetic units. This is called genetic engineering. This term is clear. This is known as genetic engineering. So I want to give you an example. See, when criminals, okay, in the cases of criminals, what actually the police do. Okay, so they use these technologies so that they can find out the printout, right, by the help of the DNA, right, because of the fingerprint we can easily find out the DNA, right. So these all the things will come under the genetic engineering. Is it clear? So let us look about the aims of genetic engineering. I have already cleared, but let us learn more. Okay, so the main aim of genetic engineering is to transplant the distributed chain by general nucleotide chain. Okay, means it is we have to distribute the chain by general nucleotide chain. Is it clear? Now alternating the phenotype of the organisms according to will by adding or by removing or by repairing or a part of genetic material. Means we can add, we can remove or we can cut or we can do any type of changes in our genetic material. Is it clear? Yes, that is known as genetic engineering. Okay, the removal and the addition of the thing. Okay, that is known as genetic engineering. Now to produce bacteria, plants and animal with desired characters. So we having a if for example we having a simple character, but what we can do by the help of genetic engineering, we can add, remove, cut or we can have many numbers of characters in the same species, right? Yes. Now it is also helpful to develop genetically modified organisms, right? Means if any type of organisms, so we can also modify that organism. Now next we having to develop genetically modified food. We can also modify the food which we are eating, right? So by the help of genetic engineering we can modify organisms, we can modify food or even we can find out the fingerprints by this technology. Okay, Now, next we having techniques of genetic engineering. So what is the techniques? So firstly, we having two steps. That is, for in first step, isolation and study of the properties of desire. Okay, so properties desire genetic material through which changes are created in the structure and functions of the gene. Means we can having the different types of functions, so that functions we can add it in the genes, right? Okay, or and also in the genetic material. Okay, we can add any type of information in that. Okay, now next step we having discovery of the chromosomes less genetic material is known as vector. Okay, vector means the carried of genetic units. Okay, those who carry the genetic units, right? So that molecules such as plasmid, cosmids, lambs, faces, and also the viruses. Okay, so what is the technique? The technique of isolation and purification of DNA have been successfully developed. H.G. Coranen in 1968 has really described the artificial synthesis of genes of known sequence of the nucleotide. Means he have discovered so that we can conclude as a vector we can carry the genetic units. Is it clear? Yes. So I have already told that by the process of transformation, right, transduction, manipulation, etc. Okay, so by this the process of genetic engineering, chemical synthesis, transplantation and heterogeneous DNA, and transduction, autoproduction, and the observation of polynucleotide changes possible. So these all the terms I'll show you in the example of DNA. Is it clear? Now next we having recombinant DNA technologies. Okay, so what is recombination DNA technologies? It refers to the joining together of DNA molecules from different species that are inserted into a host organism to produce new genetic recombination. Means in our DNA we can add any other type of genetic information or any different type of DNA molecule in the place of also in host organisms also, so that host organism can produce new genetic recombination. Means we can create any type of combination by removing or by adding the molecules. Is it clear? Yes, this is the meaning of recombinant DNA technology, right? Recombinant means again we have to combine with other molecules, that is known as recombinant. Okay, now recombinant DNA technology involve the transfer of specific genes. Okay, so by this technique what is happening? Transfer of specific genes, right? So from one organisms to other by using restriction, endonuclease enzymes and appropriate vectors. Means by the help of these vectors we can transfer from one organism to another. Okay, now next we having tools of recombinations DNA technology. Okay, so what are these tools are? So following biotechnology tools are required for recombination DNA technology. Firstly we having foreign DNA, next enzymes, and third is our cloning vectors. Okay, so what is foreign DNA? Means the DNA which is transported from one place to another, that is known as foreign DNA, right? So the DNA segment to be transported from one organisms or by vector molecules, it is called as foreign DNA. Okay, so in that foreign DNA we having different types. Okay, we having following types. So first of all we having complementary DNA. So that complementary DNA we can also relate with cDNA. C means complementary. Okay, so what is complementary DNA? Okay, so DNA which is synthesized by mRNA. mRNA means messenger RNA, right? Those who send message from one generation or one genetic to the other, right, which carry the message. Okay, so synthesize on mRNA template with the help of enzyme, that is reverse transcriptase, that is called cDNA, means complementary DNA. Okay, so what is the main process? Okay, means how does it look? So DNA strands formed in this way is that isolated from RNA to DNA complex. Okay, RNA to DNA complex, and this isolated DNA strand synthesizes its complementary strand by enzyme DNA polymerase and also become double stranded. So I'll show you. See here, I have drawn this diagram. Okay, so you just see, this is mRNA right here. This I have written by pencil. So this is mRNA, means messenger RNA. So this is a single number of RNA. Okay, inside this there is lots of information, right? So what is the next process? Firstly, it is a single strand DNA, means it will send a single strand DNA to the next process. Okay, and after completing the full process, means complementary, means the combination of that message of the DNA. So the next step we having complementary DNA, means the single strand will form a double helix structure. Okay, means it will convert it into a DNA. Is it clear for you? Yes. So this is the example of complementary DNA. Now nextly we having synthetic DNA, that is sDNA. So what is synthetic? It is artificially synthesized in the laboratory from free nucleotide, that is triphosphate, with the help of DNA polymerase. Means by the help of DNA polymerase we can easily do this work in our laboratory. Is it clear? Yes, that is known as synthetic DNA. Okay, now next we having random DNA. So what is random? It is the entire DNA, means it is a full, entire means full DNA of an organism which is broken into a small fragment with the help of restriction endonuclease enzyme. Okay, means it is a full DNA. Okay, in that DNA. Okay, so we having small broken fragments. Okay, means places, so that we can also fix another DNA with this. Okay, we having a combination. Okay, so that is known as random DNA. Now the role of enzymes. Okay, so recombinant DNA technology involve the following steps. So first we having lysing enzymes. So what is lysing? Means enzymes which is used to open the cell or dissolve the cell wall are called the lysing enzyme, means the enzyme which can dissolve. Okay, that is known as lysing enzyme. Now next we having cleaving enzyme. What is cleaving? Means if there any space is present, that is no, or another enzymes will fixed on it, that is known as cleaving enzyme. So these enzymes are used for the cleavage of DNA molecules. Okay, so it is also of three type. Okay, so that is exonuclease, endonuclease and restricted nuclease. Okay, so what is exo? These enzymes cut off DNA, means here you can see in this structure. Okay, so there is place so that we can fix another DNA with the help of the endonuclease. Okay, so these enzymes cut off nucleoids from the 5 and 3rd end of the DNA molecules. Means you have seen the structure of DNA, right? So on the one end there is 5 end present and on the other there is a 3rd end is present. So by the help of this exonuclease we can combine different types of DNA, right? Now endonuclease. These enzymes cut off DNA molecules at any point but not at the ends. Means they can cut but not on the end, means not in the end. Is it clear? Now restriction endonuclease. Restriction endonuclease means, see here in this diagram. Okay, in this picture, see, this is the full DNA, that is called enzyme cut both the DNA strand by the same site, right? So what is the main function of restricted DNA? So restricted DNA can take place on the group. right? So that the foreign DNA can combine with that DNA. Is it clear for you? Yes, means these enzymes cleave DNA fragment of a chromosome into a small DNA fragment, that is bearing one or few genes at a specific point, and that is called the recoagulation sites. Is it clear? Yes. Now next we having synthesizing enzyme. Okay, so synthesizing means these enzymes help in the synthesis of the DNA, that is DNA polymerase. We having the example, right? Now ligase. So what is ligase? These enzymes join or seal the gap present between the DNA, means in the DNA if there is any gap present, so that gap is fulfilled by the ligases. Okay, now alkaline phosphates. Okay, so these enzymes act on the phosphate group from 5 to the DNA strand, means one coil is of 5 strand, right, and another is of 3 strand. So these enzymes act on the phosphate group of 5 of the DNA strand. Now next we having cloning vector. What is cloning vector? It is a small piece of DNA that can be stably maintained in the organisms and into which a foreign DNA fragmentate can be inserted by cloning process, means any type of any foreign molecule or any foreign thing that can be connected with the DNA, that is known as cloning vector. Is it clear? Yes. Now nextly we having some characteristics of an ideal vector. So right, so what is the characteristics? It has the capacity of duplication. Duplication means a single bond or a double helix structure, that is known as duplication, right? 2 bond structure. So its molecular weight will be less, so that it can be easily joined with genes with a large and a highly molecular weight, right? And isolation and purification of vector should be easy, right? In this means in this process, isolation is easy, and also it should not be degraded inside the host. For example, we having plasmid, virus and cosmids of the ferments, right? So what is plasmids? Plasmids is also a small, plasmid is a small extra chromosomal DNA molecule, that is also genetic material found in the bacteria, means in this bacteria genetic materials is present. So it is a smaller than bacterial chromosome, and it having the capacity of self replication. Okay, now nextly we having viruses. So some virus exhibit ligogenetically life cycle in the host. So it is a type of cycle, so that in that cycle it plays as a host cell role. Okay, and these are not harmful for the host cell. Okay. Now next we having cosmids, right? So what is cosmids? Cosmids are a small sized cloning vector. These are also a small cloning vector, and it produce cohesive or necked end with the 310 of the restriction enzymes. Okay, so they also create a restriction of the enzymes. So these three are the characteristics of the ideal vector, right? Means a plasmid, virus and cosmids. Okay, so in next class we will continue with the next topic. Thank you.